Olaf is one of the leading scientists in studying the brain as a network, understanding network properties of the brain. And many of you know about the Human Connectome Project, which is a generously funded program to understand the network properties of the brain. Well, the word connectome, I didn't know where it came from. So I asked Olaf, and he said, he made it up. So this is the guy who made up the name connectome. Olaf, let's see. Olaf did his undergraduate work at the University of Tübingen in Germany, and then went on to Rockefeller University, where he worked with Gerald Edelman. And that initial work was in biochemistry. So he was cutting up brains and looking at chemical property and biochemistry of the brain. But his real love was mathematics. And then the book, Drummelhart and Feller, on parallel distributed processing, which was the beginning of neural networks, came out, and he was intrigued. And he moved over to a mathematical perspective. So his dissertation was on synthetic neural modeling, computer simulations of perceptual and motor systems. And then one more little tidbit is he went on to do a fellowship, again with Edelman, at the Neuroscience Institute that Edelman set up in La Jolla, California. And two other fellows in that group in the early 90s were Carl Friston and Julia Turner. And so that was a pretty august company. Now, they were all unknowns at that time, including Olaf. He was unknown too. But since then, they are now really the leaders in computational neuroscience using functional imaging. So I'm not going to take any more of Olaf's precious time. We can get started.
Very much, Jim, for inviting me, for organizing this visit, and everybody who I've been meeting with this morning. It's been enormously stimulating. I'm almost losing my voice. It's been so much great conversation, and I'm sure I'll revisit some of the highlights of the things I've talked to you about this morning or this afternoon in the course of my talk. So as Jim said, I was trained as a biochemist, and so this has a little bit more than just autobiographical meaning. I'll show you this first picture here. Okay, what is that? It's not a bowl of spaghetti. It's not a brain. But it is actually, that made a difference there, right? It is actually a, what is it? It's a molecule, right? And as a student of biochemistry, I was intrigued at the time by protein structure and by the fact that you could glean so much information about what proteins did, where they resided in the cell, what their properties were, how they interacted with other molecules, by looking at their molecular structure, at their three-dimensional arrangement. It turns out that proteins, like many other systems that we study, are actually networks, and that we can reconstruct and represent many complex systems in the form of network representations as collections of elements and their interconnections. And that's particularly true, I think, of the brain, and that's been the approach that I have been pushing on for the past more than 2 decades now. Networks are mathematical objects, primarily. They are collections of nodes and edges. The relation between nodes and edges define their topology. They're encountered in many different types of systems, social systems, a political block. On the left, you can imagine what the colors red and blue stand for, and a biological network interacting proteins and their interactions in the yeast cell, shown on the right. They have made inroads in many different disciplines, are used as models to represent complex systems and their structure and interactions across the board. And we are also now catching up in neuroscience, using network models more and more often. And I want to give you a little overview in my talk as to what this work is all about and some of the things that we've been working on in my lab. This figure here may seem oddly familiar to you, but it is not the figure coming from the Churchill and Lisinowski classic article from a quarter century ago, it's actually a representation of different ways of constructing network data in neurobiological contexts, all the way from interactome networks in the lower left-hand part of the scale that record very fast and very short time scale interactions among molecules, all the way to systems that reside in the upper right hand of this diagram, which are social networks, interactions among individuals and social groups that we can study with behavioral and online methodology. In between are all the different gradations of scale and observation methods that allow us to access relational data from neurobiological systems, the anatomy of circuits, of whole brain architectures, the dynamics of activity unfolding on these circuits that we can access with electrophysiology, optical recording, and neuroimaging. All these different methodologies at different levels of scale in both time and space allow us to construct networks of interacting elements that move through time that we can study with network neuroscience methods. Constructing brain networks from observational data it's in itself a laborious and difficult and time-consuming process. And this diagram here just gives you a very brief overview of how that's done. Shown here with the example of the human brain. But just as a reminder, we can actually construct networks from animal model systems as well. C. elegans, Drosophila, the rodent brain, and the non-human primate shown above. We'll come back to those a little later. And there's two main avenues by which we can extract network data from brain systems. On the left side, what we illustrate here is the pipeline or processing procedure for extracting structural or anatomical data. In an imaging context, this is typically now done with diffusion imaging and tractography, where we try to infer putative fiber pathways going through the white matter on the basis of diffusion signals coming from the brain. The end product of that is a matrix, essentially a lookup table of what's connected to what. This is the connectome that Jim referred to, the term we coined about 15, actually more like 12 years ago. Just to tell you an anecdote, when I Googled the term connectome back then, I wanted to be sure nobody else had thought of it. I got exactly 9 hits, and none of them related to brain. One of them, I remember, was to a dating site, connectomy.org, which unfortunately is long out of business. Now you Google connectome and you get 250,000 hits or something like that. It's been, for whatever it's worth, it's been quite invasive as a concept. So that's a matrix of connections, pairwise relationships that represent anatomical connections between bits of brain that may be brain regions or voxels that we summarize in the form of a matrix, in this case a symmetric, undirected, and weighted matrix of relationships. That's a mathematical object that we can directly use as a basis of graph theory and network science modeling endeavors. On the right side of this diagram, totally different way of looking at the brain dynamically through activity and activity patterns that change through time. This gives us time series observations, perhaps coming from EEG, MEG, or from fMRI, from which we can then construct, with a variety of time series analysis tools, a set of, again, dyadic pairwise relationships that can be as simple as a cross correlation. Quite often, that's the measure that's used in resting state fMRI analyses, to which I return throughout the talk later on. So that gives us another matrix, in this case a matrix that summarizes the covariance structure of the dynamics. It's a covariance matrix, and that we can analyze as a functional network. And jointly, structural and functional connectivity can be studied using the tools and methodologies of network science and connectomics. A few words about what is common among them and what distinguishes them, because this is often forgotten, I think. Yes, both can be represented, in fact, in a common space of nodes as dyadic relationships or edges. They have topology, often multi-scale, and often they also have modularity, to which I will come back a little later, what that means. But there's also differences. Structural connectivity is physical, material in nature. It defines a sparse network of relationships that are synaptic projections or connections among neurons. It has complex attributes such as density and weight of connectivity, physiology, It changes across time, but we think by and large, especially at the macro scale, relatively slowly. And it shapes the flow of information and allows information to flow in certain ways, but not others. Functional connectivity is very different. It's a statistical description of dependencies among often remote brain regions. This is how it was defined originally by Carl Friston back in the 90s. And it is much more dynamic in nature. It changes on much faster timescales, we think. And it visits and revisits a very large number of configurations, even in the course of resting state, as I'll come back later on. So it is the outcome and the end result, if you wish, of neuronal communication events that occur in the underlying anatomy. Very different animals in terms of the way they are measured, the way they are described. And one point I want to make throughout the talk is that we have to keep these distinctions in mind between both structural and functional connectivity when we actually want to analyze these data in a way that is commensurate with the nature of where the data comes from. Here are some of the descriptive network tools that have been deployed very widely. Simple nodal measures such as how many connections are attached to a given node degree. The clustering coefficient, a measure of how cliquish the neighborhood is around a node, how many of my neighbors are neighbors of each other. Motifs or subgraphs that tell us something about elementary processes perhaps occurring within the network. And then the fundamental concept of distance, which defines a minimal set of steps that must be taken, that cannot be, that must be taken in order for influence or information to pass from one node to another. Most networks we study in our field are not fully connected, but they're sparsely connected, and so not all pairwise relationships exist. This means that if two nodes want to influence each other or pass information, in many cases this has to go through intermediate steps. The distance, the path length, tells us something about how proximal they are with each other. This is very important for functional connectivity, it turns out. Two more concepts that are fundamental. One is community structure. It's something that people study in networks quite a lot. How can I decompose a large network into a set of blocks or components that are densely connected within, but only weakly connected between each other? That partitioning also imposes upon the network or allows us to identify nodes that are critical for communication. I think you will agree that the node that's shown in black in this diagram on the lower left is critical for passing information between these two communities. And if it is lost, these two communities can no longer communicate with each other. So there are some elements of networks that we can identify using network science tools that are privileged in terms of the capacity they have, at least the capacity for passing information across different parts of the network. And finally, highly connected nodes, which have many connections, are rich in that setting, can also be highly connected to each other, more so than expected by chance. And that's a phenomenon called rich club architecture, coming from the social network literature, as you can imagine, which is often found, but not always in biological networks, to which I will also return later on. Okay, now network tools that of the kind I've just described are widely used in the field now in neuroimaging and in other aspects of neuroscience. They're descriptive. They allow us to compile statistics about how a network is structured in a descriptive mode. But there's a lot more going on in this field now that I think is increasingly called network neuroscience, that it goes beyond just describing features of networks. It goes into areas such as tracking how connectivity changes across time, for example, in resting state, so-called dynamic networks, looking at modularity, communities and networks on multiple scales, also in resting state and anatomy, accounting for the important role of space in allowing certain network structures to exist and not others, generative models that allow us to answer questions like from what statistical distribution is this network drawn, models of dynamics on top of networks that allow us to highlight communication events and signaling, controllability, if I want to move a network from one state to another, what is the minimum amount of energy I have to inject, and network morphospace, which is an embedding of different networks in a low-dimensional space to allow their comparison and mutual relationship. These are all aspects of network neuroscience that are being pursued. Like I said, it's not just about describing, not just about measuring clustering coefficients or path lengths anymore. It's also about asking questions about why networks look the way they do and how they function dynamically. So first I'll say a few words about connectomes across different species, the data that we have in hand so far and what they have shown us about commonalities and differences between the brain networks of invertebrates and vertebrate species across the board. Then a few words about the human connectome and a few aspects of modular organization and the importance of hubs and ridge clubs in network functioning. Then a few words about modeling of communication processes, what they tell us about how brain networks are communicating, and finally, the thorny subject of dynamic functional connectivity, how connectivity is changing across time in fMRI resting state. First, a few words about multiple species. We have actually in hand now a handful, not many, but a handful of data sets coming from a few invertebrates like C. elegans and Drosophila and other vertebrate species that we can now take a look at with network science tools and graph theory tools. And we can ask questions as to what are common features or what are features that differ across these different animal species. The work in this area, connectomics, in some ways started with the work of a group at Cambridge, which laboriously reconstructed the brain of C. elegans, about 300 neurons, fairly stereotypically wired. This is the only wiring diagram we currently possess of any nervous system at the cellular level of detail that is considered to be quite complete. It's about 3 to 4% connected, so only about 3 to 4% of all connections actually do exist. It's been revisited over and over again in recent years as new graph theory methods and tools have become available. People have been going back to this data set over and over again and analyzed and reanalyzed it over and over. A few of the themes that have come up, there's a fairly uneven, unequal degree distribution. There are some neurons which have a lot more connections than others. These are putative hub regions. There's high clustering and short path length. And there's evidence for modular organization, some set of cells being more densely connected to each other than expected by chance. And we have also evidence for highly connected neurons in this nervous system being preferentially connected to each other. This is the so-called ridge club organization I mentioned earlier. These are the features that have been discerned so far in this particular nervous system of a fairly simple organism. Moving along the scale here to Drosophila, this is work that I've done with An-Chin Chiang's group a few years ago now, looking at the mesoscale or macroscale, if you wish, organization of the Drosophila nervous system organized into 47 or 49 local processing units. These are sort of brain regions in Drosophila brain space, and their directed connections amongst each other. This was gleaned from individually labeled, fluorescently labeled neurons that were put into a common template, a brain template, aggregated across many individuals, and density and directionality of connections were computed from that data set. We're seeing modular organization, we're seeing highly connected LPUs, we're seeing a tendency between highly connected parts of the Drosophila brain to also be highly connected to each other. Once again, that feature of ridge club organization that we see play out also in this nervous system. Moving to the rodent brain, similar features. Once again, you're now noticing I'm starting to repeat myself. This is the mouse connectome, which was produced in a seminal paper by the group at the Allen Institute in 2014, and has been analyzed by Mika Rubinov and colleagues. Subsequently, modular organization, highly connected nodes that are also interconnected amongst each other, an unequal degree distribution, so not every brain region in the mouse has the same number of connections, but there are some that have more than the average, much more than the average. And again, common features of organization that I mentioned earlier, modularity and ridge club organization among them. Moving on to the rat, this is work I've done in collaboration with Mikael Botha, Larry Swanson, most recently Joel Hahn at USC. Larry Swanson, of course, extremely well known for his work in the rat nervous system as an anatomist. And here, Swanson and colleagues have collated, based on expert curation of the literature, a complete picture of the cerebral cortex, 73, I believe, regions, recently upgraded to 79, with a couple of thousand or so directed connections among them. Many of the connections that you see in this matrix are based on multiple reports in the anatomical literature, so they're fairly solid information about their existence and strength. And again, we're finding, if we analyze this network diagram using network science tools, evidence for high clustering, short path length, modularity, that is, the network can be broken down into components, 4 of them in this case, that are more cohesively connected with each other. And highly connected brain regions, of which there is a handful here, are also highly connected to each other. If we map these modules you just saw that we derive from the topology of the connection diagram back onto the flat map of the rat brain shown here in Larry Swanson's flat map layout, you'll see that these modules, 4 of them colored differently, are actually spatially contiguous. So there's a pleasing analogy here between, on the one side, geometric proximity, and on the other side, topology and topological density of connections between them. Finally, the non-human primate, which has been studied by us and others for a number of years now. In fact, that was a matrix that we had available to us back in 1991 when I first started using graph theory approaches in this type of data. This is a more complete rendering of the macaque cerebral cortex, 242 regions. Many of these regions with high degree, with many connections attached, and they also interconnected amongst each other. I'm showing here the spatial layout of where those highly connected regions are located. If you look at the anatomical labels up at the top, there is regions that reside in lateral prefrontal cortex and cingulate cortex, inferior temporal cortex, medial parietal cortex, and other regions, cingulate cortex, that are known to be highly connected from a number of anatomical reports. And the point is they're also connected to each other, more densely than expected by chance, forming this well-known motif by now of witch club organization. So there are many common properties, to sum up what I just talked about, that are shared across different nervous systems of different species. There's a typically broad degree distribution, meaning that we find that there's a handful of neurons or brain regions that have many more connections than the rest of the brain. Every one of those regions or neurons has its own characteristic connection profile that we believe has something to do with its functionality. There's high clustering and short path length, modules that can be discerned using modularity detection algorithms and that are interconnected with putative hub nodes, and the tendency for highly connected nodes to also be highly connected to each other, which is something that is variously called a witch club or a clique or a central or core periphery architecture. That's a type of organization that I think is very intriguing because it makes some predictions as to the structural substrate, the anatomical substrate of where information might become integrated in such a nervous system. Why are there so many common features that we observe in nervous systems as distinct as that of a worm and a non-human primate? And later on, you'll see the same features pop up in the human brain. In some ways, it's puzzling because we do not yet have any features. We've never found any features in the, let's say, the primate brain that are not shared in at least a qualitative way in the brains of much simpler organisms. It looks like every organism we've looked at has a modular architecture, highly connected network elements that are also mutually interconnected in a witch club or core periphery organization. And there's several reasons, I think, that are driving this. We've been spending quite a bit of time working on and attempting to discern the evolutionary pressures that give rise to this common architectural distribution. And one important factor is space. As has been noted by none other than Ramon y Cajal, one of the organizing principles why neurons, in his case neurons, in our case circuits and networks, look the way they do is because there's an overriding limit or constraint on space, on material, on material resources that are needed to construct neurons and their connections, on volume that is in finite supply, obviously, in any nervous system, which ultimately leads to a very high premium on making connections short and connecting brain elements that are close to each other in space. And conversely, put a very high penalty on anything that is long distance and remote. Those connections are costly. They shouldn't be there if all that mattered was conservation of cost and energy. And that last fact is a hint as to why perhaps those connections do exist, because we think they confer a particular adaptive value. Here's a schematic diagram of a model, if you want to call it, or an idea, a framework that Ed Bullmore and I proposed some years ago now about how we might understand the common topology we see across many different nervous systems, including the human brain. And it goes sort of like this. If the brain was only built around the constraint that it has to be cheap, it would look like a lattice. Not a lettuce, not the vegetable, a lattice, okay? A lattice, and that's shown here on the left. Connections would be optimally short, but as you can see, that would put a high price on communication. You would need a lot of steps to get from one end of the brain to the other. That's bad for many reasons. It takes up time. The information is transformed along the way. There's potentially noise corrupting it, etc. We don't want that type of brain for functional reasons, but from a cheapness perspective, that's the cheapest brain you can have if you want to have any connections. On the other side is the random topology, which perhaps ironically is the most transmissive with respect to getting information from one place to another, but it's also the most costly because the wiring that needs to be expended on a random brain is astronomically high. Somebody once computed that for the human brain, they have the same number of neurons and the same degree of connectivity. It would need to be 10 kilometers in diameter to accommodate the amount of wiring that would be needed if it was actually a random architecture. So we can't walk around with that. There's no point. So that's out. Lattice is out for functional reasons. We think that the solution is somewhere in the middle. It's an engineering problem where we have two different constraints that have to be jointly optimized. Low cost, high performance. It's a trade-off. And different topological motifs, different types of architecture promote one and the other. And so what we actually see in the topology of circuits that we have studied so far is the imprint of these two forces, opposing forces working against each other, coming to an agreement somewhere in the middle of the scale. Yes, there's cost minimization, so wiring is not unnecessarily long, but there are some features of cost that have been expended. Some cost has been expended on features of the topology that confer particular functionality. And that is in the service of, we think, adaptive processing and cognitive performance. So far, hypothesis. We have done an extensive set of modeling studies that I have no time to review, including generative models that allow us to gauge the importance of these two factors in the human brain, for example, but a framework that we still consider to be somewhat of value. Now a few words about the human brain. When I first started getting interested in the brain, it was largely the domain of neurologists and neurosurgeons in medical schools. And in fact, the anatomy of the brain was largely contained in atlases that showed pictures like this. It's an actual photograph of a dissected, of a brain that's been cut open and where the where a few white matter tracts have been exposed. So these are long bundles of axons, myelinated, that go between different parts of the brain. This is the white matter architecture visualized in a photograph. And only fairly recently do we have the technology to actually visualize, estimate, infer likely trajectories of white matter tracts in the human brain using non-invasive imaging. This is diffusion imaging and tractography. You've all seen pictures like this. They even have graced album covers of bands, et cetera, et cetera. And there's been a whole connectome project, of course, devoted to understanding this particular pattern of wiring. The great advantage of this non-invasive technology is that we can also study people, of course, that are alive and well. We can study their behavior and cognition in relation to features of their neuroanatomy. In our first study with Patrick Hagman, now almost 10 years ago, which, by the way, was conducted on 5 subjects, something that we could never get published today. I was reminiscing about this with somebody this morning, that things were possible 10 years ago that are no longer possible. Anyway, we looked at the diffusion, the inferred topology from diffusion imaging and tractography in 5 subjects in a network of 1,000 randomly parcellated nodes across the cortex. Once again, found features that you have heard me repeatedly state earlier about other species nervous systems. Unique fingerprints. Each bit of brain, each parcel has its own unique connectivity pattern of incoming and outgoing connections. With diffusion imaging, we can't tell what the direction of the connections is, so I have to hedge my bets a little bit of whether they're incoming and outgoing, but they're connections to other regions or other parcels. And this is important because we can actually infer for regions that we don't know what their function is, we can actually infer something about their functionality based on who they talk to. This is routinely done in protein interaction networks. If you have a protein you don't know the function of, you ask what is it associated with, and then based on similarity with proteins that you do know the function of, you can infer the function of this unknown protein. Same in bits of brain. Connectivity in a network is very informative about what the functional contribution and activation patterns of that region will be. Degree distributions are broad, perhaps exponential, probably not scale-free, but at least broad with a heavy tail distribution. So there are some bits of brain in cortex that have more connections than others, many more connections than others. Putative hub regions. There's high clustering and short path length, the hallmarks of small world topology. We can break this structural network down into modules that are somewhat spatially coherent, but are densely connected within and weakly connected between. And the interlinking hub regions, those high density, high degree nodes that I've mentioned earlier, also have a tendency to connect to each other with connectivity that's denser than expected by chance. We called it at the time a structural core, and only later on did we hit upon the concept of Rich Club organization through my colleague Martin Mannenheuvel, my collaborator, who started that line of work with us. Just to reiterate what this actually is and what this means, there's a schematic diagram of our network on the left where the gray shaded areas refer to structural modules. These are sets of nodes that are more densely connected within, and there's relatively few connections between these modules. The important nodes are those in black. They are ensuring that information can actually flow relatively freely between these different communities. Now in the middle organization, what I've done is I've added a few extra connections between those central hub regions shown on the left in black. And this now allows the opportunity for those regions to also share information with each other. This is the essence of a rich club, a so-called rich club because the members of the rich club are rich because they have many connections. It's not about money in this case, although that often goes together in social systems. They have many connections and they're also mutually interconnected. So they're perhaps in a social setting maximizing their influence over the rest of the system. This mode of organization is found in the global air transportation system. It is not found in protein networks, for example, interestingly. So it's not universal to every network. It's not trivial in that sense. It may or may not exist in a given complex network organization. In the context of the human brain or brains in general, it occurred to us that this is actually of potential interest if you're interested in communication patterns. If you want to create a short, efficient path from one part of the brain to another, from one part of this network that you see in the middle, in one module to another module, the only way that you can do that is by accessing a hub node, one of those red nodes, and skipping across the rich club and getting to the other module and connecting onwards there. This is analogous to how we plot long-distance air travel. We have a local leg, then we go on the big transatlantic, then we have another local leg at the other side. And we think of this scheme as a potential substrate for information integration, for integrating information that is shared and first accumulated and then shared within this red subgraph that you see here, the so-called rich club. So Martin Mannenheuvel and I set out to look for this in the human brain. In fact, that's where we first got started with rich club organization analyses. And to make a long story short, in our very first study, we found evidence for not only highly connected nodes, this has been shown before, but also for these highly connected nodes to have more connections between them, and often long-distance connections, than expected by chance, where chance is defined as a null model that preserves the degree distribution of the network, but rewires its global topology. So the null model choice kind of is important here. Rich club organization is present, if we want to communicate from one end of the brain to another, and we're deliberately picking any possible combinations of nodes and of sources and targets here, almost 2/3 of the time do we have to actually travel through the Ridge Club. Not only a Ridge Club node, a member of the Ridge Club, but actually 2 Ridge Club nodes that are linked by a Ridge Club edge. So this part of the network, which comprises a total of about 10% of the brain, is accessed disproportionately much if we are interested in communicating fast and efficiently through the system. It is a little bit like a long distance, I use a metaphor here, a highway system that connects big cities to each other, where a lot of the long distance travel aggregates. If we damage or impair Ridge Club nodes or edges, we can do this computationally in a network setting. The integrity of communication is disproportionately affected. This sets up a hypothesis that, in fact, damage to hub nodes and connections between hub nodes is particularly predictive of brain disorders, something that has been tested in numerous contexts, including a meta-analysis shown here on the left by Nick Crossley and co-workers a few years ago, where they've gone through the literature on something like over 30, I think 36 or so brain disorders of different types. You can see the list here, everything from Parkinson's disease to autism, schizophrenia, Alzheimer's, depression, post-traumatic stress, etc. And asked the question based on a large number, over 20,000, of structural scans in brains of patients, where does the disease take hold first? What is the part of the brain that is affected, atrophied, impaired based on structural scans in those disorders? And there's a strong statistical bias towards that impairment occurring in regions that we would, on anatomical grounds with connectome analysis, predict are hub regions, highly connected hubs in the human brain. So it looks as if many disorders have something in common, a signature here that says if hub regions are impaired, there's a disorder. And that is certainly one small bit of evidence here to point in the direction of hub nodes being important also for the healthy functioning of the brain. Now, I've only talked about anatomy so far. Maybe you've noticed I've not even mentioned time courses or functional connectivity yet at all. And yet, many of you are working with fMRI and are really more interested perhaps in the dynamic patterns of activity that unfold either during rest or when we drive the brain with stimulation or impose an external task. This very evocative movie on the right, which I've seen the first time 10 years ago now, Justin Vincent's work with Mark Rakel, is a projection onto the brain surface of a resting state scan of a healthy adult doing nothing particular in the scanner. And the point I want to make with that is that it's on the surface of it certainly quite dynamic, changing, and the activation peaks that you see here shown in orange are of the same order of magnitude as the ones that we would evoke with a task. So the key question for us in the lab has been for many years, we wanted to understand a little bit better of how patterns of dependency that we see in functional connectivity, how those patterns are shaped by the underlying anatomy and by communication events that unfold within the anatomical skeleton. So the question is, how do we get from the left side of the picture to the right? This is a forward modeling or generative model question. Our approach, well, first a few words about resting state networks. This is a high-level review for many of you. How do we get those resting state networks? We take frames from a movie like this. We essentially compute cross correlations, cluster or rearrange the nodes in a certain way so that coherent, cohesive assemblies of regions are blocked together. And we can then partition this network, and these blocks that you see along the main diagonal of highly coherent, functionally connected sets of regions have now gotten names such as the default mode network, the saliency network, the frontoparietal control network, the visual network, etc. So these are now components. We can map them back onto the brain surface where they occupy reproducible, highly conserved positions across individuals, and there have been a number of large N studies of mapping these types of networks, some by Thomas Yao and others by Jonathan Power from the Peterson lab. So we have now in hand a sort of a taxonomy, if you wish, of different subnetworks that have reliably stable anatomical locations are coherently activated during rest, and are also, it turns out, coherently activated in many, many task studies as well. So where do these functional networks come from? What is the underlying generative model that can give rise to these coherent blocks of functionally coupled brain regions? This is, I think, a very important question. If you're studying functional connectivity, I think at some point you have to ask, well, where is it coming from? What is shaping, what is accounting for the dependencies and the blockiness, the modularity structure I see in this functional connectivity? And the approach that we've taken, starting now more than 10 years ago, is to implement dynamic systems, dynamic neural mass models that replicate or generate synthetic fMRI responses. There's two main ingredients to these models. Starting at the bottom, there's differential equations that describe what individual neural elements are doing across time, excitation, inhibition, conductances, things like that. Those are parameterized based on empirical observations. So even though you see many parameters here, actually most of them are totally fixed. We never change them. They define what individual neural populations are doing, how they're operating, when they are driven by input. On the top, you see a coupling structure. That's a connectome matrix, a coupling matrix in this case that is binary. And we then end up with, at each brain region, with a node which has 2 or 3 equations of motion, and we have couplings between them. So that's a coupled system of ordinary differential equations. It's actually something that's very familiar to physicists and people who study computational neuroscience. We can integrate these equations, we can simulate the activity ongoing in the system, we can pass this whole thing through a synthetic fMRI mechanism, much cheaper than running an actual imaging center, I guarantee you, and we can come back with bold responses that we can analyze in the same way as people do in fMRI research. For example, computing functional connectivity that you see here for a large portion of the macaque cerebral cortex, including most visual and some other motor regions. At the time we did this study, we actually didn't have empirical data to compare to, so we were very pleased years later to compare to empirical data acquired by Yasushi Miyashita in his lab at the University of Tokyo. We found that our simulation, despite the fact that there was no parameter optimization at all, gave a fairly, I would say, decent account of the patterns that were seen in the empirical data that we obtained years later. This line of research has since been taken forward by a number of research groups here and in other parts of the world, and it's even given rise to a simulation software package called the Virtual Brain that you can download for free that allows you to do these simulations on data sets of your own, acquisition perhaps, and make predictions and analyses of these generative models in the context of functional connectivity. So that direction of elaborating biophysical models more and more has been taken on by others. We were going the other direction and making models that are even simpler, much simpler. In this case, just a linear combination of 4 different graph theoretic metrics we can compute directly on a connectome. I think it was the path length, Euclidean distance between nodes, as well as a couple of more difficult measures to explain searchability and transitivity. And if we do this and we combine these 4 graph descriptors together, the only parameters here are 4 beta values essentially, we can come up with a very good prediction of functional connectivity. In this diamond-shaped diagram, on the left part of the diamond is an empirical fMRI recording coming from the work by Hagman et al. And on the right side of the diamond, we see a prediction based on structural connectivity from the same individuals, and on top of that, a graph theoretic model, which runs in MATLAB in under 1 second. So it's a model that can predict functional connectivity based on anatomy in a relatively simple, straightforward fashion without even requiring any dynamic simulation. A few of the diagrams on the right show you how well we do with seed points, with comparing empirical and modeled functional connectivity patterns across the brain. We're doing a lot better for parts of the brain that we believe are more accurately captured by diffusion imaging and spectrography. Another type of model we've worked on in the lab is a so-called spreading model that comes from social sciences, has been studied in the 70s when people became interested in understanding the spreading of innovations or perhaps riots in crowds of people. And it's a model where a given node adopts the opinion or the coloring of its neighbors if a certain threshold is reached and a certain number of its neighbors have changed affiliation. So it's a spreading, sort of viral spreading model that is extremely simple in the way it's formulated. It's not meant to be biophysically motivated. We wanted to use it as a tool to understand something about the topology of the human brain. Releasing a couple of different seeds, colored red and blue, unfortunately, across this human brain network, we asked the question, where in the brain, on average, do these two wave fronts of viral spreading collide? Why? Because those are parts of the brain where information comes together. You're spreading information from the red node, you're spreading information from the blue node. The topology defines where the connections, where the spreading events actually occur. And at some point, the two fronts of the spreading process come to a halt because they clash into each other. Where is that happening on average if we take all pairs of possible perturbations? It turns out that we are finding a spatial paradigm, which you see on the lower left, that is perhaps evocative of known distributions of association in higher cognitive regions across the cortical surface. These are examples of three different neural mass, graph theory, and spreading models that we have used for trying to predict communication events and patterns of functional connectivity in a generative mode. Just to give you, end this section of the talk with an overview of the framework here. So the thinking is like this. We're starting with an anatomical network. That's our wiring diagram. It defines the possible ways in which information can be transmitted. I underscore possible because not all of these ways may be realized at any given time. But we do know, I think we do know, that information cannot spread in the absence of anatomical connectivity. The anatomy in some ways defines a space of possible communication events. Then a dynamic model, whether it's a spreading model or a neural mass differential equation model or some other model of interacting neurons or interacting brain regions, will produce a set of spreading events, signals flowing through the anatomical connections, which then give rise to functional connectivity. What you measure with the bold response is, in part, the outcome of a collective underlying reality of communication events or signaling events unfolding in the anatomy. Now, the important thing is to realize is we can measure the network on the left with diffusion imaging spectrography or some other method. We can also measure things on the right. We can certainly do fMRI, cross correlations, or some other time series measurement, which gives us a network of dependencies. But we actually don't have access to what's in the middle. We actually do not see the communication events themselves. Instead, they become accessible to us, we think, through modeling, for example, of the communication processes on complex networks that we can implement in various ways. And I have a suspicion that this middle, somewhat elusive level of causal signal flow, information flow through the network is somewhat synonymous with what is often talked about in the field as effective connectivity. Finally, a few words about dynamic functional connectivity. How did we get on that topic? We actually came to it through our analyses of simulation data. In the macaque system I showed you earlier, working with Chris Honey over 10 years ago, we made a little movie that was deliberately meant to be an analog to the Vincent movie I showed you earlier. That movie is colorful, much better than this. This is black and white, and the parcels are pretty big, so it's not as pretty. But it shows you what a simulated bold response during a spontaneous activity event in a macaque monkey might look like. And if you take those time courses to make a correlation matrix, you get functional connectivity from that. It did occur to us that there were fluctuations along the way, and we actually windowed these simulated data at the time, computed functional connectivity for each window, and looked at the changing properties of the resulting functional networks across time. We used for our metric not correlation, but transfer entropy, which was applied to the underlying fast neural activity, not the bold response. And it turns out that regions that were identified as hub regions, such as area V4 and area 46 in the prefrontal cortex, residing from the overall average of functional connectivity, showed significant fluctuations along the way. So the hubness of a hub region, if you wish, fluctuated across time. A hub like area 46 that we identified based on the anatomy and the predicted functional connectivity really wasn't a hub at all times. It was a hub more so at some times than at other times. We call this phenomenon sort of hub dynamics. Keep in mind, this is done in a simulation. So we cannot blame these fluctuations on motion artifacts or scanner noise or limited samples, because it's actually fast voltages that go into this that have 30,000 time points each. So it is a, quote, real phenomenon, at least in the simulation itself. To physicists, this is not a surprise at all. You can get additional time scales, in this case, slow time scales, slow fluctuations, from a system of coupled differential equations where those time scales are not built in. They are simply emerging from the dynamics that occurs that has metastability, intermittency in its phenomenology. Others have taken this line of work much further than we have since, including especially the group of Gustavo Decco, Victor Yerza, and others who have written about and shown beautiful examples of multistability and near criticality in simulations such as this one. Strangely enough, I showed this movie and our findings at the time to people in the fMRI community back in 2007. And nobody, well, either saw the significance of it potentially or wanted to believe there was any fluctuations like that going on. Now, more recently, the topic of dynamic functional connectivity has become quite exciting. It's a little bit like Game of Thrones. It's a lot going on in this arena, and there's some dragons involved too, which are your reviewers. People have shown over the past few years that in fact there are fluctuations that you can discern with sliding window approaches, such as Katie Chang's classic paper here with Gary Glover, that we can approach the topic of fluctuations either edge by edge or looking at network states using clustering approaches pioneered by Vince Calhoun. And there have already been a number of studies that have related dynamic fluctuations in functional connectivity to levels of consciousness, psychiatric disorders, developmental stages, and ongoing cognition. And there's a profusion of, there's a lot of work. There's a profusion of work out there that has reported on these fluctuations in functional connectivity across time in the resting state as well as in task activations. But we have to be careful. As it turns out, not everything you see in a windowed cross-correlation pattern is due to actual changes in state underneath. And so we started wading into this arena somewhat cautiously with the work of Vic Betzel, my former grad student, who had the following insight, that there was an interaction between how much fluctuation in a connection across time you would expect to see due to noise, due to just uncertain estimates over shorter and shorter time windows, in relation to what that correlation was when it was estimated over the entire period of time. Little thought experiment: if your correlation over 10 minutes between two brain regions is 1.0, which is about as high as it can get, obviously, then the correlation must be 1.0 along the way. Okay, it has no degree of freedom. It cannot be low and high and then come out 1.0 at the end. It has to be 1. It is a narrow envelope, in fact, no envelope in this case, of variation that we would actually expect to see. If your correlation error is 0, and over the long time scale, you can get to 0 many different ways. You can get to 0 by being 0 all the way along the way, or you can get to 0 by being high, low, high, low, high, low, high, low, and then you get to 0. Okay, it's almost like an entropy argument. There's many ways to get to 0. There's only one way to get to 1. And so you have different envelopes of experience, of expected fluctuations across time that you can actually compute, and against which you can evaluate how do my edges actually fluctuate empirically. And what Rick then constructed is a statistical approach to look at what he called excursions, edges that were unexpectedly strong or weak along the way. It turns out most of the time edges are exactly where you would expect them to be based on chance alone, but some of the time edges exceed or undershoot the envelope of expectation. And in fact, those events, they bundle together in time. There's times when many edges are outside of their statistical norm. And he called these events excursion events because they're unexpected. And when you look at these excursion events and you aggregate them together, you look at many of them across time and you kind of average them. They have common topology, or common topography actually. They have a similar appearance. They look similar to each other. They often involve, in fact, the disconnection of task positive from task negative regions, default mode regions, and regions involved in cognitive control and external stimulation. While when edges are fluctuating more or less expected by chance, non-excursion events, we're seeing much flatter connection profiles, less extreme in the expression of connectivity magnitude, and actually it turns out also much less stereotypic. This line of work has been taken forward by my postdoc Makoto Fukushima in recent couple of years now. We've been struggling mightily to get this work into literature, but so far, well, we might be close now. He's been looking at fluctuations in modularity. Modularity is something, remember, we can actually objectively measure, extract from functional connectivity matrices. And he's sort of built time series of modularity across time and constructed an envelope, a statistical model, fairly conservative, within which fluctuations of modularity should occur. Again, looked for the extreme events of modularity being either extremely high or extremely low, and then stratified his data, his time points, based on high modularity expected or medium modularity and low modularity states. If we aggregate these states together, we're finding that they have, again, recognizable topography. The high modularity states shown up at the top at the left, the low modularity states on the right, intermediate in the middle. If we do k-means clustering on our data, analogous to what Vince Calhoun and others pioneered earlier, and we choose a k of 2, we sort of retrieve our high and low modularity states. Same with a PCA decomposition, the difference between the high and low modularity state is retrieved. And it turns out that our thinking now is like this. What we're seeing is fluctuations in modularity that are actually statistically reliable and exceed what we expect by chance. That those fluctuations account for a lot of the temporal variability that has been observed in the past and have been observed also by others. For example, it was Russ Poldrack and Max Schein in their recent work in Neuron. And we've done a ton of other work, including looking into different data sets, HCP data, as well as the NKI sample, different postulations. We're seeing individual differences in the propensity to which people engage in high or low modularity states that are reproducible across multiple imaging sessions. And we also see relationships to the underlying structural anatomy. When we have low modularity expression, those functional connectivity patterns are much more closely related to structural connectivity than when we see high modularity states. The high modularity states are states when the brain is breaking out from the anatomy and assumes a state that is actually less well predicted by anatomical connections than what happens during low modularity events. We've been on this topic for a couple of years now, but have had some issues with getting this work out, and soon I'm sure it'll appear. I think it speaks to the sort of a theme that is now going through a number of papers, I think, in the dynamic functional connectivity literature, namely that there is an ongoing alteration or perhaps oscillation between states of the brain where brain systems are more well delineated or segregated from each other, have higher internal coherence and less connectivity between, to other states of the brain that are perhaps more integrated in the sense that there's more connections between communities than there are necessarily during those high modularity states. That going back and forth between high segregation, low segregation events, we think accounts for a lot what goes on in dynamic fluctuations in functional connectivity estimated by Bold. And it's unfolding on a skeleton, anatomical skeleton, which has ridge club and hub regions, which begs the interesting question of what actually triggers those switches or those transitions from high to low modularity, and whether or not highly connected regions of the brain have a special role to play in that process. I have a lot of people to thank, obviously, and I would not be, my talk would not be complete without advertising a new journal I started last year. I'd have nothing to do, obviously, so let's start a journal, which is really positioned at the intersection of network science approaches to the brain and neuroscience. It's published by MIT Press, fully open access. The first 2 issues are out. You can look them up online. And we obviously welcome you reading those articles and perhaps submitting yourself in the future if your interest is in that intersection of network science and the brain. Thank you very much. Question?
Back to some of the raw anatomy for a second. So, obviously, if you look across species, we know things like raw neuron size or brain size or number of neurons is not terribly predictive of, what we think of as cognitive functions, right, on a continuum. But I'm kind of intrigued by some of those numbers you showed earlier with percentage of possible connections, kind of seemingly going up with species. Am I reading too much into that? Do you think that's a good predictor of cognitive ability?
I think I wouldn't read too much into those particular factors. Why? Because keep in mind the elegance, for instance, that's a cellular network, individual neurons and their connections, 300 of them. If we did a density estimate on a mammalian brain at that level of scale, our density would be way below 3%, I would predict. It will be much, much more sparsely connected. In fact, we sort of know what the density should be for a human brain. It's about 1 in a million of all possible connections are realized. So that's a very, very sparse connectivity pattern. Density of these networks is unfortunately quite relative to the level of scale at which they're measured and how you aggregate your data into regions and projections between them, and also where you make a cutoff. There has been some work out there that suggests that the mammalian brain is very, very densely connected. This is only true if you count every axon to be a pathway, and that's maybe not a prudent way of proceeding, given that there is false positives as well as false negatives in the data, and an axon or a few axons do not really have the same status as 100,000 passing between regions. So the density, unfortunately, is quite dependent upon what level of resolution your data are acquired, and I wouldn't necessarily draw conclusions from that. So to do real comparative analyses will require ultimately some homogeneous way of acquiring data, some way of acquiring data that is somehow uniformly applied. And perhaps this can be done within at least a mammalian range of species where we have diffusion imaging and spectrography available to us, including on post-mortem fixed tissue that is, it can be imaged over very long periods of time with very high spatial resolution. So that's work to be done still, but yeah.
So this change between segregation and integration, you're talking about at the end, how much of that is accounted for by this very large-scale segregation between the default network and the active network? How much can be attributed to other modular segregation? What scales do you see this?
It does seem to be the case that it is largely driven by that major disconnection between these sort of more extrinsically oriented parts of the brain and the more intrinsically oriented. I forget, some people actually made a taxonomy of this recently. So that's driving it, it looks like. That's the major disconnect, if you wish, that gives rise to this more modular configuration and more segregated configuration that we're seeing. We're also seeing, I think I mentioned it, if we take many examples of those high modular states, they are mutually more similar to each other. They fall into a more closely connected class than those flat states that are integrated and resemble the anatomy more, but are actually more heterogeneous amongst each other. So we are still trying to understand what all this means. In the work by Shine et al., there was a relation to pupillary dilation and neuromodulatory input, which connects this, I think, in a pleasing way to a mechanistic process that might actually bring about these switches. We did not track this here. We don't have access to that data from the Connectome project database. But whether it's an active process of coupling and decoupling and what triggers it, or whether it's a pure fluctuation randomly driven, is perhaps still open, but an intriguing question, I think.
Looking at the images, it seems that there's mostly connections between cortex to cortex connections. And so I was wondering, is that just an artifact of the sample, or is it a difference between subcortical structures?
Yes, it's a good question, and I don't mean to come across as entirely corticocentric. However, what makes the cortex more or less appealing from this point of view, from this type of analysis, is that it's connections within a single system. It's very difficult to do, for instance, good diffusion imaging spectrography involving the cerebellum. The structure is quite small. It has, you get anatomically implausible connections if you track down there and you don't do this well. It's a part of the brain we don't capture very well with spectrography at this point. Only experts can really handle that part of the brain well. Subcortical regions, we have sometimes included them, sometimes not. And there's reasons for why we do so that have more to do with consistency of the data and conclusions than with neglecting their importance. Obviously, they are very important. In a region like the thalamus, for instance, we know has many, many subdivisions. They generally are somewhat below the resolution, the spatial resolution of the voxels that we're dealing with. And so we're getting things here that are truly artifactual, such as the thalamus projects to everything, which is not true. It's got subdivisions that are quite specific in their projections, but we just don't see them because they're too small. So it would introduce a big artifact in our connection matrix if we treated the thalamus as a single node.
Do you think what will be the future? I mean, you would think that we'd want to really know what those are doing in terms of network hubs.
Ultimately, there's no good answer here except higher spatial resolution, greater accuracy in tracking, perhaps combination with animal data that can guide or can produce some priors, expectations about what connections should be present or not, maybe from the non-human primate. So there is, we're limited right now in our spatial resolution and our ability to reconstruct accurately. I didn't say this in the talk, but diffusion imaging spectrography has, of course, it's simply a computational procedure, right? We're not directly seeing these tracts. We are inferring the most likely configuration of these tracts based on a signal that has to be modeled, essentially. And there are certainly people in the field that are making careers just, trying to build better, more reliable, more valid spectrography algorithms. So even the pictures I've shown you for sure have false positives and false negatives within them. In some cases, we know where they are even. So it's incomplete, it's still gappy data, it's still work in progress in some ways. But with especially higher spatial resolution and more accurate tracking procedures or tracking algorithms, I think ultimately we're going to get to the point where we can include these smaller, more, in some ways, more complex subcortical structures in greater detail. Yep.
Yes, I'm not sure if psychologist, so I'm not a neuroscientist. But following your talk, it struck me that two-thirds of the way through, you're making this kind of argument for the functional connectivity to be pretty strictly linked to the spatial connectivity. And then in the final third of the talk, when you were talking about these hub dynamics, it's like
it's fluctuating.
Sort of abandoning that, or contextualizing that that's true some of the time.
Very good, very good. Yes.
And then with the hub dynamics, basically, you struck me as opening the door to psychological processes being responsible for the reorganization of the way kinds of, I mean, are you thinking of those in terms of basic attentional processes, perceptual processes, memory processes? I mean, what role does learning have?
Yeah.
Because then you have the opportunity to conduct research and
Absolutely.
experiments to tease apart when do you get that kind of structure.
These are very, very good points you're making. So let me try to revisit that a little bit with a little bit more caution, perhaps. The amount of variance explained by a generative model based on the anatomy is not 100%. It is on the order of 30, 40% in our best attempts. So we're not explaining all of the variance that we see in a long-term sample of functional connectivity. This is a static picture of a 10-minute movie, if you wish. So there's other factors at play, we think, although perhaps the anatomy is not perfect. So there is more going on than just the anatomy driving functional connectivity, even at that long time scale. When you get to the shorter time scale, simple fact of nonlinear dynamics, if you have a certain system of equations, you can get those additional time scales of fluctuations that are relatively slow and have to do with metastability and waxing and waning of global states. You can get those for free. They emerge from the dynamics themselves. You actually do not have to invoke any additional active switching or attentional processes. After all, these are simulations. They don't have attention, we don't think, or a psychological inner workings. Having said that, certainly attentional fluctuations of global state of alertness and things like that play into functional activity in a massive way. In fact, this can be shown in experiments where these parameters or these processes are manipulated. So just to backtrack a little bit, these simulations are not meant to give a complete or all-encompassing picture of what goes on. They are more existence proofs that you can get structure that is resembling empirical data using a very simple, mechanistically quite simple implementation of a dynamic system. On top of that, of course, we believe, anybody believes that there have to be other active processes ongoing that further modulate these dynamic patterns across time. This has to happen, obviously it does. Still, I would say, even with that said, not all possible fluctuations are possible. So for me, the anatomy is sort of this big thing that excludes, it's like I think of it sometimes as my skeleton, my bones, right? That allow me to perform certain movements and prefer certain configurations of bodily movements over others that are perhaps also theoretically possible, but they're excluded because my joints attach in a certain way. I can't move that way, okay? So just like that, the anatomy is like a skeleton that massively constrains what's possible, but that space of what's possible is still large. And so this is not to say that the anatomy determines, in any trivial way, what is actually unfolding dynamically. It constrains. I like the word constrain. I'll say it over and over again. But then that allows still for a lot of very interesting and very important, of course, modifications of that pattern around which this evolves through time. And so that's the spirit of the models, is to not give a complete account of everything, but show what's possible with simple mechanisms and then build on that, going to more complex processes. Thank you.
What about you? I have lots more questions, but I think it's time to conclude this and thank Olaf for that really inspiring talk.